IB Bio, Option B, Biotechnology, Bioinformatics Part 8 is the second of two movies that look at B.4, Biotechnology in Medicine. Part 8, B.4b, this movie, will have its focus on the use of biotechnology in the diagnosis of disease and treatment of disease. The essential idea is biotechnology can be used in the diagnosis and treatment of disease. Here is the outline of all the movies available for Option B. Use this outline to find the movie you need for review. This movie is focused here, but this movie covers more than just treatment of disease. Part 8 examines diagnostic techniques related directly to DNA as well as the use of biotechnology in providing therapy. So let's jump right in with the first IB syllabus statement. Describe the use of PCR to detect viral infections, specifically influenza. In the way that ELISA detected small quantities of antigen or antibody in the blood, as I covered in Part 7, PCR can be used to detect the presence of pathogenic DNA or RNA. Let me say this again. ELISA diagnoses disease by the presence of the pathogen's antigens or antibodies against the antigen in the bloodstream. PCR detects the presence of DNA or RNA of the pathogen. You might say pathogen RNA? Yes, many viral pathogens are RNA viruses, retroviruses. Influenza and HIV are retroviruses, RNA viruses. The second statement, polymerase chain reaction, serves to amplify minute quantities of DNA. The use of PCR to detect viral infection provides an early diagnosis of viral infection. For people with compromised immune systems, determining the presence of the virus at an early stage of infection is critical to their treatment. A DNA virus will infect a host cell, as can be seen in this image. The DNA becomes incorporated into the host DNA. Testing host DNA for the presence of viral DNA would detect viral infection, and this can be done. Cells of the host can be sampled, and the viral DNA can be detected using PCR. Let me repeat, cells of the host can be sampled, and the viral DNA can be detected using PCR. Pay close attention in moving forward. An RNA virus, a retrovirus, will infect a host cell. The viral RNA is copied into DNA using the enzyme reverse transcriptase. The copied viral DNA is incorporated into the host cell genome. Testing host cells for the presence of viral DNA or RNA would detect viral infection. Cells of the host can be sampled, and the viral RNA or DNA can be detected using PCR. Stay tuned. Examples of RNA viruses, also known as retroviruses, are the influenza virus as well as HIV. The glycoproteins seen here on the surface of the influenza virus are the antigens that might be detected in the blood using ELISA. But remember, viruses get inside cells. They infect cells, making detection difficult. Host cells, infected cells, can be sampled for the presence of viral RNA or DNA using PCR. Remember that retrovirus RNA is copied into DNA by the enzyme reverse transcriptase. This reverse transcription happens in the cytoplasm of the infected cell, the host cell. Now, these host cells, the infected cells, can be sampled for the presence of viral RNA or DNA using PCR. Cells from the infected patient are sampled. All the RNA from the cells is extracted. A primer sequence specific to the influenza virus is added, and the primer is the key step. The primer targets base sequences specific to the virus. The primer is a probe, like a needle in the haystack. The primer binds to viral RNA only, not host cell RNA. A DNA copy is made using reverse transcriptase, but only viral RNA is copied into DNA because of the primer, and PCR amplifies the viral sequence that can be detected by gel electrophoresis. Here is a scanning electron micrograph of the influenza virus. Influenza virus is an RNA virus, so the primer targets viral RNA before reverse transcriptase copies the viral RNA into DNA. Once PCR amplifies the quantities of viral DNA, detection is possible. One more time, host cells are sampled, RNA is extracted, including RNAs of the host. The primer targets specific viral RNA, then reverse transcription copies only the viral RNA into viral DNA. Then, only then, is PCR employed to amplify the viral DNA for clear detection using gel electrophoresis. Here is the method once again. I'll let you study the details of this slide on your own. Remember that the primer is constructed to target whatever strain of influenza might be of interest, bird flu, swine flu, or other. That said, the IB syllabus focuses on PCR as the principal technique. The syllabus statement is, describe the use of PCR to detect different strains of influenza virus. You should remember the basics of PCR from your earlier study of biotechnology. You need a primer that targets the sequence of interest, the viral DNA formed following reverse transcription. For PCR, you need the nucleotides, the Taq polymerase, and the DNA sample, which in this case would be the DNA copy of the RNA viral sequence. Here are the steps in amplifying the DNA. You should be familiar with these steps from your earlier study of biotechnology. I'll let you study this slide on your own as we bring the use of PCR to detect different strains of influenza virus to an end, and we move on to the use of another technique to detect DNA sequences of pathogens. Here are the relevant IB syllabus statements. DNA microarrays can be used to test for genetic predisposition or to diagnose disease. Let's ground our understanding of the use of microarrays by defining DNA microarray. And you need to be able to analyze a simple microarray. Here is the definition with more explanation to follow. A DNA microarray, also known as a DNA chip, is a collection of short known DNA sequences attached to a solid surface. The short known sequences are the key to the use of DNA microarrays. The known sequences attached to the surface are acting as probes. They play the same role as the primer in the PCR application. Sampled DNA, extracted from patient cells, is washed across the surface. If the patient has been infected, DNA from the cell sample will bind at specific spots on the microarray, the spots where the probe sequence is from a known pathogen. The binding indicates the presence of a pathogen, in other words, is a detection of infection. Microarrays can be used to test for the expression of a large number of DNA sequences simultaneously. In other words, test for numerous pathogens that might be in the sample. Here's the basic concept of the DNA microarray. DNA probes, short known sequences of pathogenic DNA, are attached to a surface, a microarray surface. The probes would be of known pathogens, while the sampled DNA, seen here as being washed over the surface, would come from someone suspected of infection. Now, the sampled DNA, as you can see being washed, has been modified with fluorescent tags, and it's washed over the surface as such. The sampled DNA fragments will bind to the complementary probe on the microarray. If the patient was infected with a pathogen, the DNA of the pathogen would be in the sample and would bind to the probe sequence for that pathogen. Fluorescence would then occur in those locations. The technique can be started by isolating the mRNA from patient cells. All the mRNA sequences would be included, the natural mRNAs as well as those from a pathogen that had infected the cells. Now, DNA copies are made using reverse transcriptase, a primer, and fluorescent nucleotides. This creates pathogen DNA copies that fluoresce. We apply the copy DNA mixture to a microarray of short known sequences of different pathogens. Scan the microarray for fluorescence, and each fluorescent spot represents presence of a pathogen. Remember that the microarray has DNA fragments of known base sequences, including viral sequences that are all attached to a surface. Now remember, you need to be able to analyze a simple microarray. Let's work on this using another example. Starting with a patient, extract the mRNA sequences from the patient cells. All the mRNA sequences would be included in the sample, including those from a pathogen that had infected the cells. The same process would be conducted on control cells, cells known to be uninfected. The mRNA would be extracted. Reverse transcriptase using fluorescent nucleotides would create DNA copies that fluoresce. Mix the samples together, then wash the copy DNA mixture across the microarray. Remember, the microarray has short known base sequences, and these are shown in black on the diagram. The spots that fluoresce red, just red, would be unique to the patient's cells because of the red fluorescence. These are presumably from a known pathogen. The probe at the red spots would indicate which pathogen, the specific pathogen. The green fluorescent spots would represent sequences unique to the control cells. The red-green hybridized spots would represent sequences for DNA expression common to both cells, thus not unique to the patient's cells. Remember, each spot on the microarray represents a known sequence, a probe, that's attached to the surface. Based on the position of the red spot on the microarray, the spots unique to the patient, the pathogen can be identified. One more time with a twist. Extract the mRNA sequences from a patient suspected of having cancerous cells. Tumor cells express certain genes more than others, allowing for easy detection. The same process would be conducted on control cells, cells known to be uninfected. Reverse transcriptase, using fluorescent nucleotides, followed by PCR, would create DNA copies that fluoresce red from the patient cells, fluoresce green from the control cells. We mix the samples together, then we wash the copy DNA mixture across the microarray. Remember that the microarray has short known base sequences. Those spots that fluoresce red would be the sequences active from genes within the tumor cells. Red spots indicate that the patient has cancerous cells. The green spots represent sequences unique to the control cells. The red-green hybridized spots, yellow in this case, represent sequences for DNA expression common to both cells, thus not unique to the patient's cells. Based on the frequency of the red spots, the severity of the cancer can be assessed. This is our last slide on understanding microarrays and analyzing them. Go back and review the last half dozen slides to refresh your understanding. And that brings us to our next IB syllabus statement. Predisposition to a genetic disease can be detected through the presence of markers. And to ground our understanding, let's define the term marker. A genetic marker is a gene or a DNA sequence with a known location on a chromosome that can be used to identify individuals with the predisposition to develop a specific disease. Markers can be observed in the analysis of DNA blots or gels, as can be seen in this image. Markers can be observed when DNA from different samples is cut with the same restriction enzyme, then run in different lanes on a gel. Since the DNA from different individuals is cut with the same enzyme, the banding patterns will be different, and certain bands are associated with the predisposition for disease. In this slide, on the left, you can see two chromosomes, number 17 and number 13, each with different sequences that predispose women to breast cancer. The BRCA1 and BRCA2 mutations both increase a woman's risk to breast cancer and/or ovarian cancer. In the figure on the right, proteins from different alleles of the BRCA gene have been separated by gel electrophoresis. The arrows indicate the marker proteins produced by different mutations of the BRCA gene. The presence of such marker proteins that appear in a blot following electrophoresis indicates a predisposition to cancer for the individuals carrying the marker. In this image, we have a schematic diagram of the mutated allele and the normal allele along a chromosome. There are repetitive sequences unique to each chromosome just upstream from the gene. The repetitive sequences are inherited along with the gene because of their close proximity. The repetitive sequences just upstream of the disease allele are different than the repetitive sequences just upstream of the normal allele. Since the repetitive sequences differ, the cut sites differ, and the fragments produced would also be different. Thus, the analysis of a gel would show these fragment differences as different bands. Let me show you an example. Here is human DNA sample 1, allele 1, and here is DNA sample 2, allele 2. Let's say that allele 1 predisposes a person to cancer, while allele 2 does not. Due to the different repetitive sequences, the DNA in this first sample is cut at 2 sites, creating 3 bands on the gel, band X, W, and Y. The DNA of the second allele is only cut at 1 site, resulting in 2 bands on the gel, Z and Y. The extra band associated with allele 1 is the marker that would identify anyone with such a band pattern as having allele 1 as having a predisposition to cancer. Huntington's disease results from a dominant allele. If a parent has the Huntington's allele, the parent passes the allele to children with a 50% probability. A family pedigree was created among individuals from 2 families in Venezuela by Nancy Wexler. The DNA of the children was cut with a restriction enzyme, HIND3, and the fragments were run using gel electrophoresis. The family on the right in this image has no Huntington's. The father for the family on the left has Huntington's. Given that, can you see the marker that identifies the children in the family on the left as having Huntington's disease? The children whose DNA lanes are marked with a plus have a fragment that is inherited with the Huntington's allele. These bands are called markers. And so ends our look at the use of biotechnology to diagnose disease. Let's now take aim at the use of biotechnology for therapy. Here is the relevant IB syllabus statement. Biofarming uses genetically modified animals and plants to produce proteins for therapeutic use. The term biofarm is a play on words, combining the PH from the word pharmaceutical with the word farm, and the word blend, PHARM, is used in the context of transgenic organisms engineered to produce desired products. Genetically modified bacteria are used to produce insulin, factor VIII, which is important in blood clotting, growth hormone, and more. But prokaryotic cells do not carry out proper post-translational modification of mRNA. So, eukaryotic organisms are now genetically modified to produce certain therapeutic proteins. In order to create transgenic animals, the gene of interest must be inserted into the embryo so that all cells of the animal following development would carry and express the gene. You can see here that the gene for an insulin-like growth factor is being inserted just after fertilization. The transgenic pigs produce leaner muscle. This would be an example of biofarming. The transgenic DNA, including the gene of interest, is inserted into fertilized eggs. The eggs are implanted into a female animal. Offspring develop, and those expressing the gene of interest are identified. Animals M2 and M4 are transgenic and both produce the protein product. These sheep have a gene for human blood protein, factor IX in blood clotting, inserted into their cells. The protein is secreted in their milk and can be extracted from the milk. The production of factor IX from transgenic animals is an example of biofarming for therapeutic use. In this case, factor IX important for hemophilia. The offspring of this bull have a gene for human lactoferrin, HLF. The female offspring will produce HLF in their milk. Lactoferrin is one of the components of the immune system. It has antibacterial activity important for human infants. The production of lactoferrin from transgenic animals for use by humans is an example of biofarming for therapeutic use. And here is the specific IB syllabus statement. Describe antithrombin production as an example of biofarming. Thrombin is a natural enzyme in forming blood clots. Antithrombin, AT, is a small protein molecule that inactivates several enzymes related to clotting blood. Thrombin and antithrombin are antagonistic enzymes in blood clot formation. People who express low levels of antithrombin suffer from blood clots, venous thromboses, and pulmonary embolisms, possibly strokes or heart attacks. Recurring blood clots threaten people who are genetically deficient in antithrombin production. Biofarmed antithrombin provides a source of antithrombin to people who are antithrombin deficient. Biofarmed antithrombin alleviates suffering in people with antithrombin deficiency. The gene for antithrombin is linked to the lactoglobulin promoter. This allows the gene for antithrombin to be expressed only in mammary glands where milk is produced and secreted. The recombinant DNA is injected into an egg. The egg develops into offspring within the womb of a surrogate mother. The transgenic progeny are identified by PCR. The milk is collected and the antithrombin is removed from the milk. Antithrombin then is provided to patients who are antithrombin deficient, reducing the possibility of a stroke or heart attack. The gene for antithrombin is attached to goat DNA so that it will be specifically expressed in the mammary glands of the goat. The recombinant DNA is injected into an embryo, and the embryo is implanted into a female for development. Offspring are tested for the production of recombinant human antithrombin, known as RHAT. The goats that produce RHAT are bred for large-scale production of antithrombin used for therapy. Each goat can produce antithrombin for 90,000 blood donations. Here is another slide with the information on producing recombinant human antithrombin. The FDA has approved antithrombin as the first drug produced from genetically engineered animals. A human gene for the protein antithrombin is inserted into goat DNA. The modified DNA is injected into the nucleus of a fertilized goat egg. The embryo is implanted into a female. Offspring born from the modified eggs are tested for the presence of antithrombin in the milk, and those individuals are bred. Milk from the herd is filtered and purified. Each goat can produce antithrombin for 90,000 blood donations. Numerous biofarmed products are available. You can take some time to study all the proteins produced by transgenic organisms as outlined in this slide. Drug therapy for Gaucher's disease is produced by a plant. Gaucher's disease results from a genetic deficiency in the enzyme glucocerebrosidase. Protelix Biotherapeutics, a biotech company, has produced a therapeutic protein derived from transgenic plant cells. The first approved human therapeutic protein produced in plant cells is the drug for Gaucher's disease, and is produced within carrot cells that the patient drinks. And that brings us to the end of drug therapy from transgenic organisms. Now we move to our last IB syllabus statement. that examines gene therapy using viral vectors. Here are the relevant IB syllabus statements. Viral vectors can be used in gene therapy. Describe the use of viral vectors in the treatment of severe combined immunodeficiency, aka SCID. Gene therapy is the replacement of defective or missing genes with normal working genes in cells in order to correct genetic disorders. Gene therapy is the use of genetic material to manipulate a patient's cells for the treatment of an inherited or acquired disease. In this diagram, genetic material is inserted into a virus that by its nature delivers the genetic material to a cell. The virus delivers a correct copy, a working copy of the gene to an intended target cell to mitigate genetic disease. Here is a diagram that shows a working copy of a gene being inserted into a virus. The virus with the working copy of the gene is then injected into a patient, delivering the working copy of the gene into the target cell by infecting the target cell, as will be the nature of a virus. In this image, we can see that the DNA of the adenovirus has been modified with a working copy of the gene. The virus infects a cell, delivering the new gene to the host cell nucleus. One identifies the gene responsible for the disease and then inserts a correct copy into a viral genome. The virus then infects the cell of a patient. The viral genome is then incorporated into the DNA of the patient's cell. Inject genetically modified cells into the patient with the hope that they multiply. The genetically modified cells produce working proteins. The virus used as the vector is disabled or attenuated so that it is not virulent. A gene is inserted into the virus. Stem cells are removed from the patient, from the bone marrow, and then they are infected by the virus that has the working copy of the gene. Correct copies of the gene are now within stem cells. Those stem cells are injected into the patient, and working protein is produced. The genetic disease is reduced, if not eliminated. Severe combined immunodeficiency syndrome, SCID, is a deficiency of the enzyme adenosine deaminase, ADA, and it leads to the accumulation of deoxyadenosine within cells. Deoxyadenosine is toxic to T and B lymphocytes of the immune system. When immune cells are compromised, SCID results. SCID patients are unable to fight off the simplest infections. Gene therapy using viral vectors is now being used for SCID patients. Start with a bacterium carrying the plasmid with the cloned normal ADA gene. An attenuated or weakened retrovirus is used, and the ADA gene is inserted into the virus. T cells with the defective ADA gene are isolated from the patient, and the virus with the ADA gene infects those T cells with the ADA gene. Cells are then cultured to ensure that the ADA gene is active, and then the genetically modified cells are injected into the SCID patient. One more time, the stem cells are harvested from the patient's bone marrow. The normal gene is inserted into the stem cells using a viral vector, and then genetically modified stem cells are injected into the patient with the hope that they multiply. Historically, SCID sufferers were often quarantined to prevent their exposure to pathogens, but with gene therapy, children born with SCID are now able to lead normal lives. And that brings us to the end of Option B, Biotechnology, Bioinformatics, Part 8.